Today we are going to see about gene mutation. Introduction, types of gene mutation. Introduction. The change in the base sequence of gene is called gene mutation. Mutation produces an altered gene. The organism carrying the altered gene is called a mutant. The organism carrying the normal unaltered gene is called wild type. The process of producing mutation is called mutagenesis. First, spontaneous mutation. The mutation occurring naturally is called spontaneous mutation. It is due to normal cellular operations or due to random interactions with the environment. Second, induced mutation. Artificially produced mutations are called induced mutation. They are caused by certain factors called mutagens. Mutagens may be physical or chemical factors. Example, X-rays, nitrous acid, etc. Third, point mutation. When a single base pair is altered, the mutation is called point mutation. A point mutation is classified into three types, namely base substitution, base insertion, base deletion, base inversion. Base substitution. In base substitution, a base is replaced by another base. Base insertion. In base insertion, a new base is inserted. Base deletion. In base deletion, a base is missing. Base inversion. In base inversion, the base sequence is reversed. Fourth, missense mutation. Sometimes in a polypeptide chain, an amino acid is substituted by another amino acid. This is due to substitution of three bases or codon in the DNA. This mutation is called missense mutation. Fifth, temperature sensitive mutation. In some cases, substitution of base produces a protein that is active at one temperature and inactive at a higher temperature, usually 40 to 42 degrees Celsius. This mutation is called temperature sensitive mutation or TS mutation. Sixth, nonsense mutation. Sometimes mutation produces a base sequence that does not code for any amino acid. In such cases, termination of the synthesis of protein occurs at this point. This mutation is called nonsense mutation or chain termination mutation. There are three kinds of nonsense mutation. They are amber, ocher, and opal. Seventh, silent mutation. Substitution of three bases in the DNA may result in the substitution of a new amino acid in the polypeptide chain. When the substituted amino acid is closely related to the original amino acid, mutation has no detectable effect on the phenotype of the cell. This mutation is called silent mutation. Eighth, leaky mutation. When the substitution of an amino acid results in the reduction in the activity of the protein or enzyme, the mutation is called leaky mutation. For example, a bacterium carrying a leaky mutation in the enzyme will grow very slowly when the enzyme controls the synthesis of an essential substance. Ninth, transition. Transition is a point mutation where one purine base is substituted by another purine or one pyrimidine base is substituted by another pyrimidine. Example, capital G capital C pair is exchanged with an capital A capital T pair or vice versa. Transversion. Transversion is a point mutation where a purine is replaced by a pyrimidine or vice versa. Example, an capital A capital T pair is replaced by a capital T capital A or capital C capital G pair. Next, base analog mutation. Certain chemicals are similar to the bases of DNA. These chemicals are called base analogs. The base analog has the ability to pair with the base of the DNA, causing an alteration in the gene. The mutation caused by the pairing of a base analog with the base of the DNA is called base analog mutation. Hence, the base 5 bromoracyl Bu is an analog of thymine. Bu functions like thymine and can easily pair with the adenine, causing base analog mutation. Frameshift mutation. A mutation that inserts or deletes a single base will change the reading frame for the entire subsequent sequence. A change of reading frame is called frameshift mutation. When the genetic code is read in non-overlapping triplets, there are three possible ways of translating a nucleotide sequence into protein depending on the starting point. These are called reading frames. For example, the following sequence has three reading frames. DNA sequence are given below. Reading frames are given below. These possible reading frames of a DNA sequence. As the sequence of a new reading frame is completely different from the old one, the entire amino acid sequence of the protein is altered beyond the site of mutation. Thus, the function of the protein is altered. The base shift mutation is induced by acidine compounds that bind to DNA or distort the structure of the double helix, causing additional base to be incorporated or omitted during replication. Next, back mutation. The regaining of normal wild gene by mutation is called back mutation or reverse mutation or reversion. A reverse mutant is called revertant. As a result of back mutation, the original wild type phenotype is regained. Hotspot. Hotspot is a site of DNA at which the frequency of mutation is very high. Hotspots are not frequent. Some hotspots yield large deletion rather than point mutations. The mutation frequency can be very high at Me-C site. Hence, Me-C site is a hotspot. Thank you.